Okay, so another video here. This is the cells topic test foundation. This is year 10 biology and it's unit 1.1 if you've got your spec near you. So let's get cracking then. So, move him in there. So first question then, we're looking at enzymes, okay? Students are investigating the activity of enzymes at different temperatures. They set up a test tube for each temperature as shown in the diagram below. We got 10, 20, 30, 40, 50, 60. Results of investigation. Plot them onto the graph. Choose a scale. So I've gone in tens. Look, I've made it even each time. So one big square is 10. Plot the results for enzyme activity. So I've just put the crosses in. And then join in your plots with a ruler. So we're doing a join the dots. What I want you to notice, I haven't gone to the origin here, because they haven't given me data on this enzyme, so I haven't extended it at all either side, okay? Describe how it changes, so between 25 and 40. So what I did, I looked at what's happening between those temperatures. I saw that it increased up till about 40, and then between 40 and 45 we decreased again. 51, we got calculate the change between 15 and 35. Now here I've gone down to 15 and I've dotted it, found the line, gone across, and I've done the same at 35. I've then just taken those numbers, so 51 and 23, and put them in there. Finally, we're talking about controlling here. So I've given you, state the volumes of boiled lipase. Now, the reason why I've given the same amount of lipase and same amount of milk is so all I'm looking at controlling there is the enzyme activity. So I'm not changing the milk or the amount we've got. I'm just looking at the enzymes. And then finally, why has it changed? If you think if we boil an enzyme, we're going to go past that optimum temperature. So we're going to denature the active site. So it can no longer forge those connections with the substrate. So this question 1 for you. Let's look in at question 2 then. And we've got an algal cell. So this is type of plant. So we got label the cell. Well, I've got the nucleus in the middle. They've given me a large chloroplast. I know that the cytoplasm is the site where all the organelles sit. And then they've told me that I've got the cell wall. So if I've got the cell wall on the outside, I know the other thing just inside that is the membrane. Okay? I'm then going to complete the table. So the nucleus function is the control center of the cell and it contains the DNA. My chloroplast is responsible for photosynthesis. And my cell wall is structure and protection. Now, name part of the algal cell shown in the diagram which is not present in an animal cell. So we've said cell wall. We could have also given chloroplast because animal cells don't carry out photosynthesis, so they don't need them. And then last question, bit of a tough question actually for a foundation paper. Name one part of the algal cell shown in the diagram above which is not present in a bacterial cell, and the nucleus. And the reason for that is that in our form of cells, our DNA is all bounded up into chromosomes in our nucleus. In a bacterial cell, it's left floating as strands of DNA. So that's the reason why they do not have a nucleus. Question number 3 then. So complete the sentence. We're looking now that we're filling in the gaps for diffusion. So during water, osmosis moves from an era of high concentration to low. And it's through a semi-permeable membrane. Okay, and we only had to use some of the words if we notice, so I haven't used fully permeable. That's absolutely fine. Then we've got Kerry and Sajid investigated osmosis in potato chips. They set up 3 test tubes containing blackcurrant squash and water as shown. Blackcurrant squash contains sugar, and we've put a potato chip of exactly the same size. Now, if they're putting blackcurrant squash and water, they're changing H2O's concentration, okay? They're changing the water concentration in each one. State the number of tube which is the lowest concentration of water. Well, if it's the lowest, it means the most squash. And we know then that that is blackcurrant, the one with the most blackcurrant in number 3. So just move question 3 to the side. So we're in the same question here. And we've got a bit of a maths activity. They told us at the start, if we just hover back to your question 3, I'm lightsabering him now. A potato chip of exactly the same size was added to each tube. So I can put 20 in each one, okay? And then, going to see that I'm going to go to my graph to work out the mass after 30 minutes. And they told me where the bar chart is, so it's hard to do it because of the way the picture's been taken. But if I go over with my ruler, it will go to 24. So I've completed that. I've completed those 1 mark there. Complete the bar charts then. I'm just using this data. So tube 2 is exactly the same, so I've just drawn my bar in the same place. And it's the same for tube 3. It's not, I'm just going to look for 15, go and find 15 on there, and then go down. Just be careful now when you're plotting it that you realise a big square is 5. It's not 10, okay? So I nearly put 15 there. I nearly fell into that trap. But remember, 15 they've highlighted up. Okay? We've got then, state the number where the concentration of water in the chips was the same as that in the solution and give a reason. Now, our reason for that, okay, is that if it's the same, we won't get any osmosis. We won't have any water movement, which means we won't have a change in mass. So number 2, see? So no water left, the potatoes, meaning no osmosis happened. Better science would there perhaps would be occurred. And finally, explain why the potato in chip 1 gained mass. Well, our reason for that, okay, is that concentration of water outside the potato is greater than inside. This means that our water moves in, okay? So if you think in the other tubes, that squash was meaning that probably it was either the same or a higher concentration of water inside, so our movement would be out. But if we've got the natural sugars and starch in potato inside, we're decreasing that concentration of water, which then means that we're going to get osmosis coming in through the potato cell membrane. Last bit then, homework number 4. Nice 6 marker here. I really like this question and we're going to spend a bit of time going through this. So, an experiment was set up using viscin tubing as a model gut. And this is shown in the following diagram, and they've given us it here, okay? Now, viscin tubing is it's got very small holes in and it's used for osmosis practicals, okay? So it's filled with starch solution and 1% amylase. After 30 minutes, the water surrounding the viscin tubing was tested and found to contain glucose, but no starch, okay? And I just want to, amylase, and I've written it down there. So amylase's job is he turns starch to glucose, okay? So in that bag there, and that's all happening in there, okay? Explain why glucose appeared in the water, but no starch was found. Include a description of how the water was tested for glucose, and for such using iodine solution. Okay? Now, we're going to have to add on some answers here, okay? I've noticed. And I wanted to talk about this then better. Okay? So we've got, so first of all, amylase converts starch into glucose. We've said that up there. I've put that, and I'll just mark my answer as I go along, okay? Glucose molecules are small enough to pass through the viscin tubing. Glucose is outside is lower, so we're going to have it move out. Now, starch is too large, okay? Now, we've got 2 tests to do, which they've said, and I'm going to continue writing that in the video now. So we test for glucose using Benedict's, as it says in the question. A positive result is going to give us, a positive result then is going to give us a red and orange colour. And it kind of goes on a spectrum from a yellow to a red. And generally, like, the hotter the colour, if you think red seems a bit hotter than yellow, the hotter the colour, the more glucose is present. Now, we test for starch with iodine. Now, iodine, we're expecting a negative here. We've got to say the expected observations. So expected result is no change. But we do know, and that would be sort of a red-brown colour, but we do know that iodine goes blue-black for a positive. We don't need that for this question. And there you go. There's the cells topic test for you there.